John Rodakis.
Greetings to my fellow public and federal members and those watching by video. My name is John Rodakis. For the past 12 years, I've run the N of 1 Autism Research Foundation to identify and fund potential breakthroughs in ASD at many of the country's and world's top institutions. I'm also father to one of the purest souls I've ever met. Every day inspires me to be a better person and to find breakthroughs for him and the millions of other children like him. I have a degree in molecular biology and spent much of my career in medical venture capital. I'd like to share two priorities I hope we can explore further in later meetings. First, I'd like to advocate for a significant broadening of NIH's biomedical research efforts in ASD beyond genetics. While genetics can be very important in ASD, the vast majority of individuals, some estimates as high as 80%, have no identifiable genetic anomaly. This despite the fact that we now have over 1,000 potential autism risk genes. At this point, the evidence is overwhelming. There is more at work than simply genetics. But the vast majority of research funding, both public and private, remains concentrated there. I would like to ask for a deliberate, meaningful, and measurable effort to broaden NIH's research agenda into other high potential domains, such as the immune system, the microbiome, and mitochondrial cell metabolism. This isn't about replacing genetics, but I believe that by broadening our lens, we will discover breakthroughs applicable to the other 80%. A second priority likely falls to the purview of the FDA. At present, there is not a single approved treatment for the core symptoms of autism. I believe part of this failure comes from how we approach clinical trials, but it's fixable. Conventional clinical trials work well in homogeneous populations, but are poorly suited to the highly heterogeneous conditions like autism. Some may say it's a failure of the treatments themselves, but time and again in autism clinical trials, we see a familiar pattern where a subset of patients show a strong, meaningful response to a treatment. However, because that response isn't broad enough across the group, the trial fails to achieve statistical significance. As a result, promising treatments, often with favorable safety profiles, never reach the patients who could benefit from them. I believe this is solvable. We've seen the FDA address similar challenges in other heterogeneous conditions, including cancer, through more adaptive and flexible trial designs. My hope is that we can convene a working group with the FDA to explore trial design that can better account for autism heterogeneity to help bring safe and effective treatments to patients who will respond to them. Thank you.